So we can slowly start and people will keep joining, I guess. Let me share my screen for a second before we actually start the session. I will verbally say what I want to say. Before we start, I would like to, because there are new settings and new meetings, and that's why we have a new kind of Zoom account, which I'm still figuring out. That's why there are some issues. Yeah. So what I would like to say before we start is to actually, I can share it on Zulip, and we will soon share it also on Twitter and wherever you follow us. I would like to invite you to save the date because on September 9th to 11th of 2024, we're going to have our first conference, the first Iceverse conference. And it's basically the idea is to bring the community closer to the users. So everyone that follows the community meetings is more than relevant, I think. And we will have lots of sessions and workshops that they are Iceverse related, but not only, and companies. So I will repeat, it's going to be also in the, I will try to edit the video at some point and add the slide. So it's going to be on the recording on YouTube soon. 9th to 11th of September, it's going to be our first Iceverse conference. So now, having said that, I would like to invite and talk about Constantin, who is our speaker today. He's a postdoc right now in Wolfgang Huber group in EMBL in Heidelberg. And today he's going to present LEMUR, which is a package for differential expression analysis in multi-condition single-cell RNA-seq data. So, Constantin, thank you for coming, and the floor is yours. Please try to share, and I can approve that we can see your slides. And then you can start.
Thank you. Let's see if this works. I haven't done this in a while, but I guess the training from COVID. So, can you see my slides?
Yes, perfect.
Excellent.
Perfect.
Perfect. Yes. First of all, thank you so much for inviting me. I'm very excited. So I'm by tradition much more of an R programmer than a Python person, but in the last half year, a year, I have dabbled quite a bit in Python. So I'm very excited. And so this will be mainly about the project and the algorithm behind LEMUR. But in the end, I also want to talk a bit about a very recent development where I made a PyLEMUR package, and also curious to hear some feedback from your side. But with that, let's get started. So I've been working on a project which is motivated by trying to predict the expression values in multi-condition single-cell data. And throughout this presentation, I'm going to mainly use this one data set by Zhao et al., where they looked at the glioblastoma single-cell expression data from 6 patients, which were treated either using control, so DMSO, or with a broad histone deacetylase inhibitor, panobinostat, and then also with etoposide, but I'm not going to pay much more attention to that. And the typical workflow for analyzing such a data set is that you collect the multi-condition data for 2 reasons, either because you want to find out if there are shifts in the cell type abundance, so cells in type abundance, or you want to figure out how the gene expression between similar cells across different conditions changes. And I'm going to focus on the gene expression changes. And to do so, what you would typically do, you collect the data. If you make a UMAP, you see that it's often dividing by the known covariates, which in this case are the patient ID and the treatment status, and you would run some integration method where there are plenty of those available. And then you get this kind of embedding here on the right, where the variance is being driven by the different cell types and cell states. And typically, somebody goes through the data and either using some reference data or some manual notation, figures out what each of these different clusters corresponds to and assigns some cell type. And then you would do a differential expression test for each cell type and each gene, which gives you this very long list of gene by cell type differential expression values. And the problem with this approach is that it is inherently unsatisfying that you have to divide your continuous kind of gradients into discrete clusters. We all know that biology is often varying continuously. However, here you have to divide these gradients, and you also have to upfront define the resolution at which you look for differential expression values. So you look at all the tumor cells as one big blob instead of maybe looking at a more fine-grained resolution, or at least you have to make that decision early on. So what we try to develop is an alternative approach which tries to avoid these cluster-based differential expression values. And to illustrate this, I'm using this example where we are now looking at the gene expression. This is the histone gene in the treated versus the control cells. And we can already see here the blue dots are the cells expressing this histone gene highly. And we can see that here in this black circled cell population, there's a high expression, whereas in the corresponding population of control cells, there's none of that. And what we want to get out in the end is this continuous inference where we have for each cell and each gene predicted what is the expected differential expression values that we expect to see here. So we would expect to see for this circuit population that there is upregulation under treatment of the gene expression. That's what we predict
Also
here.
nicht, dann erstmal, ich erst nachher.
There's some, okay, perfect. Yes, and to get there, what we do is that we first need to smooth away some of the short noise from the gene expression. So we want to get a more smooth kind of prediction where we use the low dimensional embedding of our cells to make this prediction. And then we want to answer the question, what is the counterfactual expression of the control cells? So what is the expression of these cells had they actually been in the treated condition? And this kind of prediction, so clearly this expression would be upregulated. Let me actually switch to a laser pointer. That might be even easier to see. We already know that this expression should be higher if they were treated, simply by looking at the corresponding cells on the other side. And then just by taking the difference between these two different states, we can generate the picture here on the right, which is giving us the differential expression values for each gene and each cell. So underlying this framework and this prediction models is a novel matrix factorization approach, which we call latent embedding multivariate regression, or short LEMUR. And the idea is basically an extension of PCA. So in PCA, the idea is that you have your large observed expression matrix Y, where you have the genes in the rows and the cells in the column. And this is already something which is probably different in Python versus R, but in my world, genes are usually in the rows. And then what you try to get is some low dimensional embedding Z, and you combine this with the matrix R, which spans some subspace in the high dimensional gene space, specifying how exactly the gene expression is going. And the clue of our method is that instead of having one fixed matrix R as a PCA, we treat R as a function of the covariates. So for each cell, we have some additional covariates, namely which patient does the cell come from, which treatment status, and we plug these covariates into R, and R then gives us the optimal subspace for that particular condition. And this, so if you now think about this more graphically, can be represented like this, where we have the cell population for the control cells here as an orange blob, and then we find the subspace R, which encapsulates or frames this expression. Now, if we have two conditions, we have two different subspaces, which are functions of these covariates. And all that is left to do is kind of mapping between these subspaces to then be able to make these predictions. What is the expression of a cell in one condition? Had it actually been in the other, by simply mapping these positions across the different cell positions. And a slightly different perspective on this whole problem comes from thinking about generalized linear models. So generalized linear models are a popular method in statistics to link the mean of the observed variables to be conditional on the covariates to some linear predictor, because linear predictors are nice because we can easily work with them, and they have with all the different theory, how to do stats and linear algebra with that. But often our data might not be actually linear, but we have, for example, logistic regression where we have to map the linear space to the space between 0 and 1. And this is achieved using an inverse link function called R, which takes this linear output and remaps it to the output space that we care about. And the analogy for LEMUR is that we have a similar link function R, which takes some linear predictions in the high dimensional space and links this to the mean of the observed covariates. And by generating these latent spaces and combining them with a low dimensional embedding of each cell. So I've now talked a lot about the function R, but I haven't actually said what it is. And statistically speaking, or mathematically, it's the exponential map on the Grassmann manifolds. But that might not actually be much more helpful for many people because it's a fairly obscure topic. however, what I want to get across here is that it's just some linear algebra, which might look a bit scary, but it's nothing too complicated. So this is something you can write as a two-line expression in R or Python to get these mapping from between the different subspaces. And the idea behind this is that we have these two subspaces, and to map them, the simplest idea would be just to take the vectors spanning the subspace and map them on top of each other. However, what we want to do is actually ignore the spanning vectors and just treat the subspaces as is, because you can imagine that there are many different ways to parameterize these subspaces, and we want to be independent of that. And this is achieved exactly by these Grassmann manifolds. That's why we work with them specifically. So I have already talked about how we can use this to make these predictions for differential expression values. And in our preprint, we applied this to analyze the gene expression changes during zebrafish embryonic development. So we found this nice data set with 16 time points between 18 hours and 48 hours. At every 2-hour interval, somebody sequenced a lot of different zebrafishes and then looked how the gene expression changes. And we were particularly interested if we could actually assess the interactions between the developmental time and the cell state within the cluster, for example, of the periderm. So we took two cells, the periderm, one at the top of this cluster, one at the bottom, then linearly interpolated the position of these cells in the latent space, projected this back on the UMAP. So this gave this almost kind of linear kind of trajectory. And then we could use this ability of LEMUR to predict the gene expression as a function of the underlying gene space and the covariates to make these smooth predictions here for each time point at each position and see these interactions where we can see that for the points here at the top, we have this decreasing expression patterns, where for the cells at the bottom, we have this increasing expression patterns. And the dots are simply the approximated gene expression values in of the cells in the region that we actually observed to see that these actually match the underlying expression patterns. However, these differential gene expression patterns are often a bit clunky to work with because you get one differential expression prediction for each gene in each cell. So you get the full dense matrix, and that's often pretty difficult to work with. So we have developed additional functionality to group together cells which show similar differential expression values. So this is our example from the beginning where we had these prediction, and here with a black circle, I've grouped together cells which show consistent differential expression values. So all these cells in this cluster here on the left, they are close together and they have similar differential expression value. So we group them into one neighborhood. And this may actually sound now a bit similar to what I said in the beginning with the cluster-based analysis. However, the important difference is that we get one neighborhood for each gene. So we get a different neighborhood which is determined by the particular underlying differential gene expression pattern for each particular gene. And we can then use that to conduct a pseudobulk differential expression test for the cells within that neighborhood to figure out if these patterns that we find there are actually statistically significant. So we applied this method now to this data set that I mentioned at the beginning with the glioblastoma data. And we looked specifically at the tumor population to figure out, are there actually interesting subpopulations? And for the LMO2 gene, which is interesting because it has been linked to a decreased length of survival with higher expression, we found this subpopulation of cells, which upon panobinostat treatment show downregulation. And we can confirm this pattern, first of all, by looking at the underlying gene expression values, but it's even more prominent when we actually look at the pseudobulk expression values, where you can see this stark downregulation of the expression for the cells within the neighborhood versus the almost unchanged expression for the cells, for all the other tumor cells. And this, of course, opens up all kind of other follow-up questions. So what is the difference between these cells and why do we have the specific subpopulation effect there? The neat thing about LEMUR is that it's a very flexible model, and I've now demonstrated already how you can use it with covariates, for example, as time or as an experimental condition, but you can also use it to analyze spatial data. So here I took a data set which was generated by Cable et al., where they sequenced the hippocampus from 4 different mice, and for each cell quantified the underlying Alzheimer plaque density at that cell. So this is shown here with this yellow curve and gradient. We can see that there's quite a bit of variation. And we identified that there is this subpopulation of neurons down here at the bottom of, if we make the UMAP, where this gene June, which is again interesting because it's part of the cascade which is regulating apoptosis induction upon the occurrence of these Alzheimer plaques in cells, that with an increase of the Alzheimer plaque density, here shown on the x-axis, the gene expression of June for the cells in this dashed circle area increases, whereas for all the other cells down here at the bottom again, there's no change, which again raises all these different interesting questions. So what is different about these neurons here at the bottom versus the other neurons, and how could this be relevant maybe for some kind of Alzheimer treatment? So we also did invest some time trying to validate that LEMUR actually works. So the first step is the integration performance, and we found that we have similar performance similar to Harmony. We also, that was quite important, validated that the prediction values, so that the prediction of the gene expression as a counterfactual are still reliable, and that our gene expression average per cell type is that our predicted expression values per cell type are actually similar to those as the observed average prediction expression values. And we also ran across many different data sets to make sure to learn how sensitive our tests are for detecting differential expression, and that our test actually controls the FDR, so that you don't get an inflated kind of results where, due to how the test is structured, you will get more false positive than you would expect. Yes, so there currently is already a bioRxiv preprint available, and we have, so in the tour of this project, which has lasted 4 years, I've worked on an R package, which is also available on Bioconductor. So if any of you wants to try this out, this is available. But probably much more interestingly to this crowd, for the last, I think, 2 weeks, I've been working on a PyLemur package, which is now, I think, ready for people to be tried out, although I'm a bit terrified. But yes, so it's already on. I've made documentation. There's a tutorial walking through you through all the different steps that are implemented, and documentation for all the functions. So here's a quick overview of how what the different state of the packages is. So I've implemented so far in PyLemur only the first 3 steps of the model, which goes up all the way up to making the predictions of the differential expression values. I have not yet implemented the neighborhood grouping and the differential expression testing, as that is a bit more complicated, and the R code is a lot more involved there. But it might be, yeah, I think this is already quite useful all the way to the prediction value, because this is the conceptual novelty of the machine learning side of the model, that you can actually make these different predictions for each cell and gene specifically. Yes, I have also written down some lessons learned during the programming. This might be interesting to maybe other people who are also considering submitting an SE-burst package, or who are interested in maybe bridging some of these. So one of the first things I did not realize was that I should actually use the cookie cutter template, and that would have made my life a lot easier in the first 5 days until somebody told me that I should actually use it. And now it has made my life a lot easier, I think. The other thing is that I tried to write my tutorial using Quarto, which is this R Markdown further development, which is supposed to support Python. However, I could not make it play nicely with swings for the documentation generation. In general, I just struggled a lot with swings, and I'm not happy with it. But that might just be my inexperience. The other challenge during the implementation that I encountered was that really, this is just an unfortunate way how this now is the culture, that in R you always have the genes in the rows, whereas in Python is in the columns. That means that everything is transposed, and that means that all my linear algebra I had to go through, I had to always think about which way around things go. But this is then, yeah, the other lesson learned, the next step, that it really helps to just feed in exactly the same data, make sure that you get one-to-one the exact same results, because that really protects you against having an additional transpose too much or too many in your code, which can easily happen. And then the last question, and that's actually something where I would like to hear some audience feedback, is that I'm struggling a bit with the Pythonic style. So I thought maybe we could make a quick vote, and maybe I have to skip some slides here. So right now so I struggle a bit, what's the best way to return the data and what's the expected kind of structure that people like to work with. So I'd like everybody, because we have these nice kind of reaction emojis that you can press at the bottom of Zoom, say what is your preferred way that like a package from the Essiverse should actually deal with the data. Should it return an N data, which just has extra field? Should I make a subclass of N data, which supports additional functions and has this? Or do you prefer the scikits learn kind of object things where we have like a linear regression thing, then we call fit on that, and then we can call predict on that, and all other things? Yeah, maybe like I did see some people clicked already. Maybe I'm going to count to 5 again, and then you can all click again so that we have a good picture. And I actually know how the community feels about these things. So 1, 2, 3, 4, 5. Please click. Okay, I think this is pretty unanimous. Everybody loves N data with extra fields.
You are in the wrong field, I think, the wrong meeting to ask this
Yeah.
thing.
This is interesting because, yeah, so in R, I went with a subclass approach of summarized experiment, which is very popular there. However, luckily, actually, this is how I went with it for now. So on the recommendation of a colleague, I did go with the N data with extra fields. So I'm very happy with that. So thank you very much for the feedback. And then I also want to, of course, acknowledge everybody in my group and thank you for your attention and feel free to ask any questions.
Perfect. Thank you so much. It was an amazing presentation and you made everything super smooth, I think, even the linear algebra and everything, like super smooth. If you want to ask questions, either raise your hand and I can call your name, or just put it on the chat and I can read it for you and then Constantin can reply. But I can start with something that I have in mind. I will give, no, it's fine. I can ask later. So Dimitri, go ahead. I don't think we can hear you.
Can you hear me now?
Yes, perfect. Yes, thank you.
Okay. Hi, Constantin. Thank you very much for the presentation. And I have like 2 questions. The first one, I remember you mentioned some low dimensionality thing in your in your model. Could you please tell what is this? How do you reduce the dimensions? And the second question is how fast, how scalable LEMUR is, because I believe it may work well on like 20,000 cells, but if we move to 100,000, how does it perform?
Excellent. Yes. So the low-dimensional subspaces, maybe I'll quickly go to the presentation, to the slides, which might be most relevant. Sorry. I like this one. So the latent subspace is just the linear subspace, which captures the variation for each condition. And condition for me is just a unique row in the design matrix. So in the simplest case, where we have a treated versus control experiment, as described for the glioblastoma, you have all the cells from the treated condition and all the cells from the control condition. And most simply speaking, I'm just fitting a separate PCA, which, in my case has, let's say, 20 or 50 dimensions to the control condition and to the treated condition. So that's the low dimensional embedding. So it's a linear embedding. And that also means that, to the second question, it is actually very scalable. The advantage of this model is that I can just use all the highly optimized approximate SVD functions or PCA implementations. Actually, also Severin, I think, was here earlier. I'm not sure, he's still there. Already has submitted a pull request to also support GPU to make it even more scalable for fitting the PCAs. But just as a reference, so you just mentioned that, so the 20,000 works easy, and that's easy on a computer. The zebrafish embryonic development data set had 700,000 cells. This is not something I think I could run on my computer, but it's something I can pretty easily run on a cluster, and it doesn't take too long because basically just needs to do like 5 different, or for however many conditions you have, PCA fits to the data. The more challenging bit, which I've gotten feedback on from people who have used it, is that the differential expression predictions that you get out, these are dense matrices. So usually you work with these sparse matrices because you can just ignore all the zeros. However, for my differential expression predictions, you get really one per each gene in each cell. So if you have 1 million cells by 20,000 genes, that object will kill you. I've not yet come up with a good solution how to, well, except just subset your genes or subsample your cells to avoid that, because I don't conceptually know what else I could do about it.
Okay, thank you very much.
Can?
Two things. One thing is, can you correct for covariates for the differential expression?
You mean correct, like adjust for additional covariates which you don't actually care about, or?
Yeah, exactly.
Yes, absolutely. So it follows this idea of linear models where you, if you, let's say, you know that there might be some sex bias or effect for this tumor data set, then you could just include this as an additional covariate in your design matrix or design formula. To now jump back all the way to the end, where I showed the syntax. Sorry, here so I'm using formulaic for the design matrix parsing. So you just provide all your different covariates, which refer to columns in your ops field. And there you can refer to as many as you like. And for the differential expression test, so this is shown here, you just compare the two levels that you actually care about. So if you want to compare condition against control, that's how you write this. And this automatically adjusts for the uninteresting covariates, like, for example, patient ID or sex or whatever hospital sequencing facility you have as well in your data.
And when you do the confirmation using pseudobike, what's the size of neighborhood where you think it still works? Because when you subset to 50 cells or something like that, likely the p-value estimated from pseudobike is not really pseudobike anymore. What's your perspective on this thing?
That's a good question. So the R package has a field or argument for the minimum neighborhood size. I think it is roughly 50, if I remember correctly, but I would need to check. To avoid exactly the kind of effect that you're talking about, that at some point there's just going to be so few cells in that there's no way they're going to be significant. I haven't really looked into it too much. So I think it's not so. From the data I've looked at, it's not super common that you have this super small group with a very strong differential expression values. So it happens that only a subset of, like, an obvious cluster shows differential expression. I haven't yet seen it that it would be so localized that only like 50 cells would show it. So that's why it hasn't, I think, come up too often for me that the algorithm tried to focus particularly on these. But it might be something interesting to investigate, or if you have a data set where you think this could be happening, that it selects like very small regions, one could play around with the settings there to nudge it into a direction of larger neighborhoods.
Thanks.
I can go next then. No. You're using, to reduce dimensionality, a linear method, which makes total sense to me. I'm not saying the opposite, but have you considered using, or have you excluded using variational autoencoders, which is very hot topic, and it's linear and non-linear, so you would expect to capture some interactions there?
Yes, I think it's interesting, and I've been trying to think about what it would actually mean, how one would go about trying to use a non-linear, like variational autoencoder method. because in some sense, the idea behind variational autoencoder is that you can get better predictions. So where like a linear method, you need to add more dimensions to improve your predictions because of potential interactions effects. With a nonlinear method, the idea is that you might be able to do this with a small dimensional space to get better predictions. I think conceptual bit where I'm just mentally struggling and I haven't quite figured this out for myself is what does it mean if I have the subspace? Because for me, the subspace is something in my head. I can think about a three-dimensional gene space, and then I have my subspace, which is like this two-dimensional plane in there. And I can kind of hand wavy say in a 20,000 dimensional gene space with a 20-dimensional subspace, that's kind of the same concept. But then if there's something nonlinear, then like is my subspace suddenly bent? Okay, like I still can kind of envision that. But how would I then do this mapping from one subspace to the other? Because this is right now the kind of, I think, elegant bit of the method that I can very easily adjust between those. And I can visualize this, or at least from in my head, kind of think geometrically about that. And I think this would get a lot more complicated. And I assume just I couldn't use some simple structure like the Grassmann manifold to describe these mapping, but would need to do something more complicated. I don't think it would be impossible. However, I think this particular idea might not play very nicely with the variational autoencoder. On the other hand, there are related ideas like CPA or SCGen where people have tried with autoencoders to also make these predictions. I think it loses a bit of the interpretability that you can actually expect these subspaces. And at least for me, I've had trouble getting them to work and make particularly good predictions. And of course, the other advantage is that there's very comparably few tuning parameters involved for my model, whereas for autoencoders, you have this whole range of different things you might need to tweak to get the best kind of values.
Mm-hmm. And you also compared with CPA already, and you have similar, if not better, right, performance?
Exactly. So just looking at the prediction performance, I get much better prediction with LEMUR. I have been in contact with the author of CPA, and because the initial version I did it very naive, I just used the default parameters, and that completely failed. And he then gave some advice on which other parameters to use. That improved it a bit, but not to the level of LEMUR, at least for me.
Yeah, that's okay, I guess, right? That's why methods are there, and also it was quite like early on the CPA.
Absolutely, yes. So the CPA has been around for a while, and yeah.
Thank you. Are there any more questions? You can put also in the chat, don't forget. And I want to mention that the challenges in the end, can you go to your last slide while people are thinking questions? Like the one that you were saying, the challenges with, yeah, great. So I want to mention in the first one that I also didn't know, but then I reached out to SCVerse and they let me know that there is the cookie cutter template. So please take home message, reach out, especially Julie, but also emails and everything. Sphinx, I suffered equally, so it's not just you. And the rest, I agree. It's a very nice slide, actually. And I wanted to mention also that in the conference, there will be workshops for that kind of, to help to kind of importing the ecosystem packages. So you were a bit unlucky because you did it before that, but you know.
Well, yes. I think, yeah, I tried to flex some of the things that I learned to Gregor already, like if there was a typo in the documentation or stuff I stumbled over. But yes, it's an experience.
Yeah, definitely. It was an experience for me too, but we're trying to make it smooth.
Yes, that's excellent. That's what I also was happy to kind of do it, because I feel like there's quite a bit of energy in the community trying to improve things.
Yeah. So I don't see any more questions, right? Dimitri, go ahead.
Hello again. I just sorry, I didn't read your paper yet, but I'm actually from what I've heard, it's sounds as a very nice tool I can use in my analysis. And my question is that I have single cell RNA-seq data from several time points, and I have several cell types there. If I understood it correctly, I can just take, let's say, I know I have motor neurons, and I have like 1, 2, and 4 days after treatment. So,
Yes.
and I can do differential expression analysis across all conditions without separating them, like day 2 versus day 1, or like day
Yes.
1 versus control, etc., etc. I can just do in one run and have like set of differentially expressed genes, correct?
Yes. So for the zebrafish example, I used splines, so that to give me some smooth kind of trajectory through the through time for the gene. For your case, if you have 3 time points, I guess splines would not work, but yes, it can linear fit to the 3 points to tell you which genes are increasing over time or decreasing or not changing. Absolutely possible. Yes, that is definitely possible. You could already do this on a pseudobulk level before. So if you would want to, or if you don't mind too much with this discretization step in the beginning, you can already use, for example, Python ecosystem, the PyD-seq methods to fit these kind of models, or also with the splines or kind of linear models through time. Or in the R world, there's of course in LEMUR, HR, DSEC to support this. And the clue of my method is that you can go beyond the analysis per cell type and identify for each gene which groups of cells actually show differential expression. So in particular, if there are some gradients between the different cell types, I think that's in particular where lemure could be helpful.
Now I see. Okay, cool. Thank you very much.
Yeah, but exactly. So if anybody tries it out or uses it or has some questions, feel free to email me, and also open GitHub issues if you spot some kind of inconsistency in the documentation or some error message or warning that you can't make sense of. It's often interesting when you release such a package to the broader public, what kind of use cases come up and what kind of things you never thought about people would use to call it. So I had quite a few of these already in R. Of course, I'm now even more terrified of the Python kind of things that I've made missed.
Yeah, so that's amazing, actually. I have shared all of your accounts and everything, I think, and they can find you also on Twitter, like an email and Zulip, I guess. And so feel free to reach out. That's one nice also take-home message. Like today is about community, I think, your presentation, which is great. Like reach out, especially for developers. The best thing you can do is to actually try to use their tools and reach out and let them know. Most of the people, they're more than happy to help, I guess. So yeah, I don't see more questions. And as I said, I have shared everything, all of his contacts. Please find him and reach out. And thank you to everyone for joining. And Constantin, thank you for this amazing talk. And see you in 2 weeks on our next community meeting.
Yeah, thank you again for inviting me, and have a good day.
Bye.
Bye.
Goodbye.